And thank you all for coming. I look forward to meeting some of you all later and getting to learn a little bit more about Carbondale and the school. As Greg mentioned, I'm an evolutionary ecologist. I really like to think about how geography and ecology interact to shape biodiversity dynamics across different spatial and temporal scales. So today, my talk is going to be in 3 parts. I'm going to start by giving you sort of a conceptual overview of how I like to think about ecology and geography in the diversification process and some of the major empirical patterns that have motivated a lot of my research and a lot of the questions I like to think about. Then I'm going to talk about a couple of past and ongoing projects that touch on those from a couple of different perspectives. And I'll wrap up by talking about some future research projects that I'd be excited to pursue here at SIU. So I want to start by thinking about the role of ecology in diversification and begin just by defining a few terms. This is probably familiar to most of the audience in this room, but diversification is the process that generates the living species we see today. It consists of 2 components: the speciation process, by which ancestral lineages differentiate and diverge into descendant species, the process of species birth, and then there's the process of species death, where species fail to persist through time and either wind up in the rock record or just invisible from the recorded history of life. And in practice, we can't actually observe this entire process. But what we can do is we can try to reconstruct it using patterns of molecular sequence variation in species alive today. And we do that by building molecular phylogenies. These are reconstructed diversification processes, and it's not immediately obvious that we can learn anything interesting about the actual diversification process from these reconstructed molecular phylogenies because they don't contain any of the extinct species. But in practice, the topology and branch lengths of these molecular phylogenies do contain some information about how quickly things were speciating or going extinct back in time. Now, these reconstructed phylogenies vary a lot in shape. They can be highly balanced, whereby for each speciation event, the number of descendant species in the 2 daughter lineages is approximately equal, but they can also be highly imbalanced, where one descendant lineage receives many more of the resultant species diversity. And it's not necessarily clear where to expect natural diversification processes to fall along this type of continuum. But one really interesting empirical pattern is that empirical phylogenies tend to be really highly imbalanced. There are a number of different examples we could use to show this. This is one from angiosperms. Angiosperms are tremendously diverse, hundreds of thousands of species. But what I find really fascinating is the sister group to all these hundreds of thousands of angiosperms is this monotypic genus Amborellia on the Pacific island of New Caledonia. Similar examples you can find in vertebrates like fishes. So polypterous fishes, this is about a small clade of 13 species of freshwater fishes in Africa. They are sister to tens of thousands of other ray fin fishes that live in freshwater and ocean habitats around the world. Now, the interesting thing is that null models of speciation and extinction can actually explain these patterns of imbalance. If we go to empirical phylogenies and calculate formal metrics of their shape, those empirical estimates of balance shown in the histogram tend to lie well outside what we would expect if that random process was just happening by chance. And so what these 2 facts mean to me is that interesting biology must be playing a really important role in generating these patterns of phylogenetic imbalance. And in my research, I really like to think about how ecological differences among different groups of organisms might be altering diversification dynamics to play a role in generating these patterns of phylogenetic imbalance. The other thing I think a lot about in my research is the geography of diversification, the geography of evolution. So I want to start again with just a couple of terms here. Here I'm showing 2 pairs of closest relatives, these toy species A and B, and C and D. And we would say that A and B are more closely related than C and D because their most recent common ancestor occurred more recently in time than the common ancestor of C and D. So the amount of evolution separating A and B is less than the amount of evolution separating C and D. The ellipses at the top, if those represent geographic ranges, we would say that A and B are allopatric because they don't co-occur, whereas C and D are sympatric because there are areas of their geographic range where they are living together. One really common pattern that we observe in nature is that closely related species tend to be uncommon. There are different examples that we can pull from. This is an example from South American ovenbirds. The x-axis here shows the time that sister species have been separated, and the y-axis shows how many pairs of species at that divergence time are sympatric. And again, for closely related sister species, sympatry is very uncommon. And so what this implies is that most speciation must be occurring in allopatry. Another really interesting pattern is that the probability of speciation scales with geographic area. We know this because we can look at these different groups of organisms on oceanic islands, and we can tabulate the number of pairs of sister species that we think speciated on those islands. And the frequency of those within island speciation events grows with increasing island area. And this is presumably reflecting something about opportunities for greater or opportunities for allopatry on larger islands. But what I find really interesting about this example is that the rate at which speciation probability scales with geographic area occurs at different rates across these different groups of organisms. And we can predict that rate by the spatial scale of genetic differentiation. So the x-axis here is a measure of how genetically differentiated populations become at short to moderate distances. And the y-axis is the smallest island on which we observe within island speciation. And so groups of organisms like snails and lizards that differentiate genetically across geographic space more readily than flying animals like bats or just highly motile animals like mammals require much larger island areas to achieve allopatry and speciate. And so these observations imply that dispersal must be playing a really critical role in this diversification process. So the way I like to think about the role of ecology and geography in diversification is through this sequence where the formation and persistence of allopatric populations is the first step, and dispersal is playing a really integral role in that process. Those isolated populations then need to differentiate both ecologically and reproductively, and it's that differentiation that allows them to complete the speciation process by coming back into sympatry, and that informing these species-rich communities, which allows them to start this cycle again. So at any given point in time, the diversification process has this ecological and geographic dimension, and periodically, there are major events in the history of life that cause some lineages to differentiate into sort of a new region of this ecogeographic theater that alters the diversification dynamics. And that's what's generating these patterns of imbalance that we observe in empirical phylogenies. And so in my research program, I like to think about this process from two different scales, sort of from the top-down phylogenetic scale on, I guess, on your right, and sort of the bottom up from the dispersal scale on the left. And in the next part of the talk, I want to touch on two past and ongoing projects that address that from those two different spatial scales. And I want to start by talking about snakes. I love snakes. Snakes are great. And I think they're really excellent systems for thinking about the role of ecology in diversification. And one of the real fundamental challenges of being a snake is how do you feed a large body through a small head? As you elongate the vertebrate body plan, your head becomes proportionally smaller relative to your body mass. So you're faced with this challenge of nourishing a heavy body through a small head. How do you do it? Snakes, of course, have solved this challenge really remarkably by evolving numerous cranial adaptations that facilitate a large gape. Most notably, the two lower halves of their jaw, they lack this mandibular symphysis, so they can operate independently, and that gives them a really wide gape. And snakes don't swallow their prey. They can actually use these two independently operating lower jaws to just walk themselves over their prey. And that's how they're able to, in some cases, perform these remarkable feats of ingestion. So snakes have been confronted with this challenge of feeding a large body through a small head, and they've solved it spectacularly. And the result we see today is a huge range of dietary items and feeding ecologies in snakes, vertebrates and invertebrates of all shapes and sizes. This is just a word cloud that's showing prey items that have been recorded in the diets of snakes. This is pulled from a database of snake diets that I maintain. And the size of the words here is sort of how frequently those items are included. And there are some usual suspects like mammals and other lizards, but there are some truly oddball things that snakes are eating. So onychophorans are this really strange, early diverging group of arthropods. They can shoot defensive glue at their enemies from their legs. You wouldn't think that that would be sort of a niche that snakes would exploit, but there are in fact species of snakes that specialize on eating only onychophorans. And in fact, they're venomous species. They're coral snakes, completely unexpected. There are also some really surprising things like turtles and crocodilians that show up in snake diets. These are heavily armored prey, very unusual shapes. You wouldn't necessarily expect snakes to be eating them. So huge range of diversity in what snakes are eating and how they're eating it. But snakes are also, from a phylogenetic perspective, just really unique lizards. So there are about 4,000 snake species, and they're phylogenetically nested within this paraphyletic group of about 8,000 quote-unquote lizard species. And so what this means is that snakes are unique in two ways. Not only are they unique in the range of feeding strategies that they can employ, they're also really unique because they're the only group of lizards that has evolved limblessness and then gone on to radiate into thousands of highly ecologically diverse species. So this phylogeny here is a cartoon version or a reduced phylogeny of snakes and lizards. Snakes are shown by that red point. And these lollipop diagrams are showing our estimate of speciation rate in this group and how it varies across phylogeny. And snakes are really exceptional in this regard. And in fact, they're the only group of limbless lizards that has become exceptional in this regard. Limblessness has evolved in lizards hundreds of times. And it's only in snakes where we see that resulting in enhanced diversification. Some other limbless groups of lizards are highlighted in the blue branches, and they're not doing anything special as regards how quickly they are speciating. And so I think this is a really great system to asking this question of whether or not the elevated speciation rates we see in snakes are associated with higher rates of diet evolution. We would predict that to be the case if a lot of the speciation events that are occurring in snakes are also associated with shifts in their trophic niche. And so this is a question I'm posing in the first project. And to get at this, I started by developing a very large database on snake diets, primarily harvested from the published literature, but also using specimens preserved in museum collections. This was a lot of work that I had. I was fortunate to find some undergrads who were also enthusiastic about snakes and could help me with this. But so we have close to 60,000 predator-prey observations from all over the world, from all major snake families that we can use to think about how rates of diet evolution vary across snakes and lizards. Then I developed a model that takes these diet data and pairs it with phylogeny and essentially tries to learn about different groups of snakes that have similar feeding modalities. So each one of these large circle packs corresponds to a set of snake species that the model has identified as having similar feeding habitats. The smaller circles inside there correspond to the individual snake species in our data set. And the colored circles inside those are the types of prey that the snakes are eating. So the model here is doing a good job of recognizing shared patterns of dietary preferences. Then taking these model-inferred trophic niche states, we can project these back onto snake phylogeny to think about the evolutionary history of how these states have changed through time. When did they first appear in snake phylogeny? And how did they change? So that's what I'm showing in this frequency distribution here. Each colored wedge corresponds to one of these trophic feeding modalities that the model has already identified. And the different colored wedges appear at different points in time when the model has estimated that those different feeding modalities have appeared in the phylogenetic history of snakes. The x-axis is time, and the y-axis corresponds to the proportion of ancestral snake lineages that are inferred by the model to have belonged to that particular feeding guild. And this inset curve is a measure of how much variation we see in among lineage diet breadth. So how different are these diets as we move from one snake lineage to the next? And during the Eocene and the Miocene, we really start to see a spike in the rate of increase in total among lineage diet breadth. This is corresponding to periods in time when the ancestors of some of the major snake groups that we observe today were appearing and diversifying. And importantly, the rate of increase during these time periods is actually happening much more rapidly than we can explain by chance. So if we do some null model permutation tests, they show that this rate of increase is happening very rapidly and can't be explained by chance, which is suggesting that a lot of those early divergences were associated with shifts in snake dietary niche. So the next thing I did, this was work with collaborators, is we wanted to see if this result of very very high rates of diet evolution in snakes held up if we paired that snake diet data set with another similar data set that incorporated the diets of other lizards, so non-snake lizards. And what I'm showing here is just a principal component ordination where we're taking all the lizards and snake diet data and we're ordinating it in a common coordinate system that's primarily describing variation in prey preferences along an axis from invertebrate prey to vertebrate prey and from aquatic prey to terrestrial prey. And this is just, again, highlighting the fact that snakes are really exceptional. They've colonized all areas of this ecospace, and lizards here are just confined to this upper left corner where they're eating terrestrial invertebrate prey. And when we pair these data again on snake and lizard phylogeny, we recover this consistent result of really exceptional rates of snake diet evolution. So again, I'm showing these lollipop diagrams. In the middle here, these are essentially two independent metrics of how quickly diets are changing over evolutionary time. And yes, there are lizards that there are lizards who have evinced high rates of diet evolution, but snakes are really exceptional in this regard. So there are a lot of interesting future directions that I think this work can take. And most importantly, there's still a lot we don't know about snake diets. And so there's real opportunities for continuing to learn about what snakes are eating using existing museum collections and also doing targeted field work to try to learn more about these unknown species. So this is pulled from our database of snake diets, and over half of the species have fewer than 10 recorded prey items. So there's still a lot we don't know. And SIU is actually a great place to do this because Field Museum of Natural History is a train right away, and it has a world-class herpetology collection. And the current curator there is also a snake fanatic. And I think would be really excited about getting students involved in some of this basic research. The other really important direction this work has to take is that there's a lot more biology to incorporate into the comparative methods. I didn't go into much detail about the actual mechanics of how this model is operating, but it makes the assumption that we can characterize each individual species by a single trophic niche. But we know that reality is more complex. Snake diets are varying over ontogeny. Juveniles are eating different things than adults in some cases, in many cases, in fact. And snakes are eating different things in different places, depending on local selection pressures, or what's available. And we really can't account for that in any of our comparative methods at this point. And that can actually be quite consequential for thinking about rates of evolution and how we make statements about ancestral ecologies. If we're missing this aspect, that has a potential to mislead what we think ancestral snakes were doing. And there's also a lot more to snake feeding than just prey identity. So we've been treating the snake feeding niche as something that we can characterize just by how frequently are you eating frogs, how frequently are you eating mammals. But the reality is that a frog that is small for one snake species could be big and bulky for another species. And so the actual sort of predator-prey allometry is very important in snake evolution. We don't yet have a good way to think about this from a model-based perspective. And so that's another really important direction that this research needs to take. So this next part of the talk is some of my more recent work, and I'm just going to shift gears a little bit, and I want to think about a different type of tree. So we've been thinking a lot about species trees, and now I want to think about trees that relate to individual genomes. So here I'm showing, you can think of this as a chromosome, and we have these trees along the chromosome. The tips of these trees represent genomes, so a copy of this chromosome in an individual, and the internal nodes of this tree would represent genomes of a pedigree ancestor. And because of sexual reproduction, the process of recombination breaks up genomes every generation into different bits and then stitches them back together. And that means that the sort of ancestral history of genomes is different for different places. So as you move along the genome, those pedigree ancestors change. And that's recorded in these structures called ancestral recombination graphs that we can think about as these trees that describe patterns of ancestry as we move from left to right along, along a genome. So in this orange region of the chromosome, for example, individuals B and C have a most recent pedigree ancestor. But as we move farther along, that relationship changes. Individuals C and D actually have a more recent common ancestor. And that's the result of this process of recombination. And the important point is that we can learn about these patterns of ancestry from genetic sequence data, from patterns of variation in genetic sequence data. And because genomes are big, it often requires many such trees like this to fully characterize ancestral patterns of ancestry in genomic data. And that makes these ARGs really rich sources of information for learning about ancestral processes. And one ancestral process that I'm really interested in is thinking about how the rate of dispersal has varied over space and time and across the genome. Because individuals live in geographic space, they tend to mate with nearby individuals. And that means that the patterns of ancestry reflect this. Nearby individuals tend to be more closely related than far away individuals. And because of that, we can learn about dispersal by interrogating these tree sequences. And often this question can be addressed by reframing it and thinking about where genetic ancestors of a sample lived at different points in time. So I'm going to tell you now about a project that I'm working on to think about how we can find out where genetic ancestors lived. And we do this by using these tree sequences together with the spatial locations of the sampled genomes. And so here is this toy tree sequence that's relating individuals 0, 1, and 2 that are in this abstract continuous plane that's meant to represent some sort of geography. And we want to try to reason about where the ancestors of these individuals were based on the locations of the present day individuals together with their pattern of ancestry as recorded in these tree sequences. And the basic intuition is that we expect nearby individuals to be more closely related to one another than far away individuals. And so the common ancestors of those nearby individuals should probably also be living near where those individuals are. So for example, this ancestor 3, which is the closest relative of individuals 0 and 2 on this middle part of the chromosome, maybe it lives somewhere in between them. And so the idea of this approach is actually for each of these ancestors to learn a function that we can give. The input to this function would be possible locations for where that ancestor lived, and then that function would return to us how much migration or how much dispersal had to occur in the history of this tree sequence for that ancestor to contribute its genetic material to its present-day descendants. And those are often parametric functions that we can calculate really quickly. So we can build up a picture that looks something like this, where for each ancestor, we ask how far did that ancestor have to move if it was in a given point in space. And so the warm colors here mean that if that particular ancestor was in that location, a lot of dispersal would have to happen to explain the patterns of ancestry that we see in the modern day individuals. Whereas if it were located in a gray region, we wouldn't have to invoke lots of dispersal and migration over the history of these tree sequences. And so we can use these surfaces to make educated guesses about where we think genetic ancestors lived. In particular, we can place them at the point on this surface that achieves the lowest sort of total migration that had to occur. So this is essentially kind of like a least squared solution. It's a heuristic solution. And because of that, we can apply it to really big data sets. And so I'm going to show you an application of it to human data because we have a lot of data in humans. So this is showing data from the Human Genome Diversity Project. Each one of these points represents a collection of genomes that were sampled somewhere in the world. And the size of those points corresponds to how many genomes were sampled there. And what I'm showing is a single tree for a particular region of human chromosome 18. And what we've done with these data is we've applied this method to try to learn where the ancestors on that particular region of the chromosome were. And then we've collapsed the temporal dimension of the genealogy and projected them into space so that the edges of that tree trace out historical migrations. And we've colored the edges by how recent or how ancient they were. Now, this is just for a single tree, but there are many thousands of trees that are required to describe patterns of ancestry across human chromosome 18. And so each one of those then records a slightly different version of the geographic history that's necessary to explain patterns of ancestry among these individual genomes. And so we can union all of these and make a similar picture, and it looks something like this. And the interpretation here is the same. Each one of these red lines is tracing out an inferred historical migration, and the color or shading of that red indicates the antiquity of that migration. And what I like about this figure is that it emphasizes two things. One, it emphasizes sort of the recency of common ancestry among all of us. So these more peachy colored lines correspond to recent historical migrations. And the background of this image is essentially awash in pink, reflecting the fact that we don't have to go back very far in the past to find a common ancestor. But it's also recording more ancient historical migrations that all of us share from when ancestors left Africa and peopled Eurasia. Now, once we have these, there are lots of interesting directions we can take to interrogate them, to learn about patterns of spatial ancestry and how they may have changed over time and over space. But one interesting direction I'm really interested in pursuing is applying these methods to other empirical systems besides humans. And some specific projects that I'm working on with collaborators right now is we're trying to use these tree sequences and this method of locating ancestors in tree sequences to try to learn about movement patterns in other species. So with folks at University of Melbourne, we're trying to understand if international shipping routes have influenced the movement and spread of insecticide resistance in Aedes aegypti mosquitoes. This is the causative agent of dengue, so this has implications for human health. And working with folks in the Fitzpatrick lab, we're also trying to use these tree sequences to learn about how land use change has affected population demography and connectivity in Massasauga rattlesnakes. This is a small species of rattlesnake that depends on seasonally flooded wet meadow, so it's quite sensitive to changes in land use cover. One of the big challenges with extending these methods beyond humans is that it's is actually building the tree sequences and inferring these ARGs. It's quite difficult to accurately infer these ARGs with moderate coverage genomic data. So these projects are still in the early stages because we just haven't been able to infer tree sequences that are of sufficient quality. But I think as we get more data and as sort of inference methods improve, these sorts of analyses will become more widely adopted and can teach us some really interesting things about dispersal. So I want to close the talk by discussing two research directions that I'm excited to pursue here at SIU. And for this first part, I want to take us back to snakes and pose this question of what can worm-eating snakes teach us about evolution? So the phylogeny here is a family level snake phylogeny. Each triangle represents a family, and the width of these triangles is proportional to how many species of snakes occur in that family. And what I find really fascinating is that this obligate diet of earthworms has arisen all over the tree of life, all over the snake tree of life, including in some truly unexpected places. So highly venomous snakes like vipers have evolved diets of exclusively earthworms in correlation with evolving small body size. And highly venomous species of elapid snakes have also evolved this obligate diet of earthworm eating. And we just wouldn't necessarily expect that in snakes that have evolved venom ostensibly as a means to take down dangerous mammalian prey. So all told, there have been at least 20 instances where an ancestral diet of vertebrates has been lost for a diet of only earthworms. And this provides a lot of phylogenetic replication, and I think allows us to ask some really interesting questions about potential genetic and ecological mechanisms that might be explaining this really fascinating phenotype. So in particular, I'm interested in using these repeated instances of convergent evolution to see if we can identify shared patterns of genetic variation that are associated with that phenotype. So this would involve identifying appropriate phylogenetic contrasts where we have an obligate vermivore closely related to a vertebrate and seeing if we can identify patterns of genetic variation that are enriched for that contrast across these different phylogenetic origins. And earthworms pose a couple of interesting challenges for snakes. So they have highly potent mucosal defenses that can gum up snakes' jaws. And also nutritionally, they're not great value. So they're essentially bags of water, and that means that they move quickly through the snake gut. And so snakes that are eating earthworms are actually feeding quite frequently. So there are sort of metabolic demands as well as prey defense circumvention demands that snakes have to meet if they are to evolve this phenotype. And so I think it's quite plausible that there are shared genomic regions that might be explaining the ability of snakes to do that. There are also many snakes that include earthworms as part of a broader diet. So one good example of this is the ringneck snake. It's quite common around here in southern Illinois, where they're eating mainly small salamanders. But if you were to run a transect west across the Ozarks, by the time you got into the eastern plains of Kansas, the populations of this snake are only eating earthworms, perhaps reflecting sort of just the profligate abundance of earthworms in prairie soils. But this provides a really interesting way to test whether or not we can identify shared genetic variants that are also explaining this eating polymorphism within species. So is there continuity of process between interspecific and intraspecific divergence? I also think this is a really interesting system for evaluating coexistence mechanisms that might explain sympatry among worm-eating snakes. So in southern Illinois, there are many species of obligate earthworm eaters that are coexisting in the same place, but we don't actually know what's explaining that, in part because we don't know enough about what these snakes are eating besides earthworms. When herpetologists open up a snake and see that it's eating an earthworm, they don't go any further. They just record earthworm. And so that's what we have. That's the data that are available. We don't know if these snakes are eating introduced earthworms. We don't know if they're eating native earthworms. We don't know if they're catching earthworms deep in their burrows or if they're catching them when they're active on the surface litter. So there's a lot of interesting sort of odd ecological dimensions to this phenotype that are worth exploring and I think would make great student projects. And I think funding plans for this would primarily target NSF in these two clusters. And I think for this project on coexistence mechanisms and sympatry, it's a potential candidate for thinking about state and foundation conservation funds. Because these snakes are eating earthworms, they have very specific sort of habitat humidity requirements, so they're quite sensitive to land use changes. And so there are interesting applications, potential land management applications as we learn more about what these snakes are eating. The other empirical dimension that I'm interested in pursuing has to do with these minute tree fungus beetles. The name says it all. These things are very small. They're only 1 to 3 millimeters, and they spend their whole lives living in the fruiting bodies of wood decay fungi. The males are often ornamented with these interesting horns. We don't know what they use them for, presumably sexual contests, but we really have no idea. So I think these are small brown beetles, but I find them very charismatic. And I think they have several interesting properties that allow us to ask interesting questions about them as well. So they spend their entire life cycle confined to fruiting bodies of wood decay fungi, and they can be found year-round in large numbers. And they're the primary sort of biotic agents that are responsible for initiating sort of this successional cycle and decomposition of these polypores. So they're also playing sort of important, if unseen, roles in forest nutrient cycling. And here in North America, there are many species. There are about 84 species, and they have varying host preferences from monophagous to polyphagous. And there is a sort of good taxonomy available for them, although it's no doubt in need of updating because it's about 50 years old at this point. And I think we can use these beetles to pose some really interesting research questions about how ecology, and in particular feeding biology, has been important in evolution and important in the speciation process. So has host use shift been an important speciation mechanism in these beetles? I think it's one really interesting direction. We know from some work on Japanese populations that we can identify cryptic population genetic structure that's associated with different fungal hosts. And presumably that is operating in other populations around the world. But we don't know the mechanism by which it's operating or how frequently. So I think we can also ask interesting questions about fitness trade-offs across fungal hosts that might be explaining some of this cryptic population genetic structure, and that might explain why host use shift is important in speciation. I think this is also another really interesting system for asking whether or not ecology, and in this case feeding biology, is predictive of within population genetic diversity or within population genetic variation. So this is sort of a long-standing puzzle in evolutionary genetics, is why is there so much genetic variation within populations? Our theoretical models suggest that this has to do with population size, but attempts to explain patterns of genetic variation in nature have met with mixed success. But I think this sets us up to make some interesting contrasts between monophagous and polyphagous beetles. So host abundance at the landscape scale is predictive of the number of coexisting seed beetles that you find within a polypore. So fungi that are more abundant in the landscape also have more species-rich seed beetle communities. So it's possible that if you are a polyphagous seed beetle, you have more habitat and more food available to attain larger census sizes. And so we might expect those larger census sizes to result in increased genetic variation within polyphagous beetles compared to monophagous beetles. But we just don't know. So there's really no molecular genetic resources available for these beetles in North America. And so one of the directions I'm interested in pursuing is sort of establishing genomic resources for these beetles that we can then use to begin addressing some of these questions. Again, targeting NSF mainly for these research questions. I think they're also a potential candidate for these two programs, advances in revisionary taxonomics and poorly known and poorly sampled taxa, because we just really have no idea about many basic aspects of these beetles. Okay, I got through that. Thanks for bearing with me. You could probably tell I was very nervous. So just thank you all for listening, and thanks to some of my mentors and collaborators with this work, and thanks to undergraduate students and graduate students who help with data collection and some of the conceptual problems that I've been wrestling with. So thanks, and I'll take any questions.